welcome back to our channel. This is Science Lectures 256. If you've not yet liked, subscribed and followed, please do so. So today we are continuing with our series of invertebrates, and today we are going to look at platyworms, or they call platyhelminthes. But before we proceed, there are some new terms that we are going to encounter. Number one, protostomes. Protostomes. So from flatworms, annelids, mollusks, echinoderms, and vertebrates, we see the development of a digestive system. And the flatworms are the first organisms to develop a digestive system. So that gives us another division. So those animals are either protostomes or what we call deuterostomes. Deuterostomes. So what are protostomes? Protostomes are animals whose blastopower results into the formation of a mouth. So in other words, they develop a mouth before the development of an anus, as compared to the deuterostomes that develop an anal opening before the development of a mouth. And these protostomes are further divided into two larger clades. Number one, we have what they call the lapotrophozoans, and the diisozoans. Diisozoans. So the lapotrophozoans are these animals that have any of these characteristics. Number one, they undergo mosaic cleavage. Then they have what we call elaphotrophora. And elaphotrophora is a horseshoe-shaped feeding structure, or they have what we call a trachophora larvae. If not a trachophora larvae, we call it ciliated larvae. If not ciliated larvae, we say larvae that has a band of cilia on it. So for any organism to be classified as a laphotrophozoan, it must undergo mosaic cleavage, have a laphotrophora, which we say is a horseshoe-shaped feeding structure, or it must have a trachophora larvae or a ciliated larvae. And under laphotrophozoa, we have members of phylum Annelida, members of phylum Mollusca, and members of phylum Platyhelminthes, or what we call the platyworms. And we look at the ecdysozoans. These ecdysozoans have two major characteristics. Number one, they have a cuticle, and this cuticle gives them the power to mold. So all organisms, all protostomes that have a cuticle and have the power to mold are ecdysozoans, and these are arthropods and nematodes. So after having seen that, I didn't want us to encounter those terms minus knowing their meaning. I want us to look at the general characteristics of flatworms. Number one, they are triploblastic. That is possession of three germ layers. That is the ectoderm, the mesoderm, and the endoderm. Number two, they undergo cephalization. We said earlier, cephalization is the development of a head. Then number three, they exhibit bilateral symmetry. So these are the first organisms we are seeing that exhibit bilateral symmetry. And we said bilateral symmetry is associated with cephalization. And this is so because cephalization, that means there is development of a brain. And where we have a brain, we have faster responses to external changes of stimuli. Then they are acylomate. So these are organisms that do not have a xylem because their xylem is filled up by mesoderm tissue called the parenchyma cells. So where you would have the xylem is filled up by parenchyma cells. Then they are either parasitic or free living. On top of that, they are either aquatic or terrestrial. So those are the general characteristics of flatworms, and we are going to see more specific ones. So after having looked at that, next we are going to look at the outer covering of flatworms. Outer covering of flatworms. So the outer covering of the skin of flatworms is made up of two things. It is either ciliated, or it has a syncytial tegument. So these ciliated organisms have cilia on the outer body surface. On top of that, this cilia is produced by certain cells called the rabidites. And this cilia, apart from covering the outer body, is also used for locomotion. Then these ones and this is very common in the free living, especially planarians. Then these ones that have a syncytial tegument, syncytial means multi-nucleated, and tegument is covering. So these ones that have a syncytial tegument are multi-nucleated. Then this means this: if I have this as my cell membrane, this cell membrane shall house very many nuclei. This is a nucleus, this is a nucleus. Basically, it means one cell membrane housing very many nuclei. And this is very essential, especially in the parasitic forms. Of flatworms, because as we shall be seeing later, it was essential for them to absorb nutrients from the host. Number two, secrete wastes from their body. And number three, secrete waste from their body and also resist digestive enzymes. So that is it for the outer body covering. Next, we look at their masticular system. So the masticular system of flatworms is made up of basically three major types of muscles. Number one, we have what they call longitudinal muscles. We have what they call the circular muscles, and we have what they call diagonistic muscles. So all these play a very vital role in locomotion. And locomotion in annelids, especially the free living, is responsible for running away from predators, hunting, because some of them are carnivores, and also looking for mates. Looking for mates. So we are still continuing. After having seen the muscular structure, we need to look at the various organs within the flatworms. So these organisms, we are starting to see the development and the cell organization. Remember, we began from cells, as we saw in sponges that have choanocytes. Then we started seeing the development of tissues within the cnidarians. Now we are starting to see the development of organs and organ systems. Let's begin with the excretory organs. These organisms also have what we call excretory organs. And the excretory organs of flatworms are made up of what we call the protonephridia. Protonephridia. Definitely, you're already guessing that the protonephridia, we are starting to see the development of what we call rudimentary kidneys or primitive kidneys. Then it's also made up of what we call the tubules, also made up of what we call the nephriopods. Nephriopods, and the phlegm cells. So what we have here is the outer structure of a phlegm cell with the tubules. So this is my nucleus. These are flagella. In fact, we have also a flagella this side. Flagella. Then this is what we call a tubule. And this is a tubule cell. And each tubule cell has its own nephriopoa. So these ones, and this from here to here is what we call a phlegm cell. So it is called a phlegm cell because this flagella at the base generates currents that look like flames of fire. That's why they are called flame cells. How does it function? So when this flagella generates currents, the currents will bring in things like excess fluids and other wastes from the body. So these wastes shall travel through the tubule and will be excreted via the nephriopoa. That is how it works, and that is the general overview of the excretory system within the annelids. Next, we look at the digestive system and their nutrition. What do they feed on? So, as I had earlier hinted, the parasitic ones, we have the parasitic ones, and you have the free-living flatworms. These ones are carnivores. They hunt. Then these ones feed on already digested food. So these ones that are free-living tend to have a blind gut. As I said earlier, these ones, we are starting to see the development of a digestive system within the animal kingdom. So these ones, the gut of this organism is considered as one of the most primitive in the animal kingdom. I said it has a blind gut. This gut is made up of a mouth, then it is made up of a pharynx. Then this mouth for the planarians is located on the ventral side. Then for that flukes, it is located on the anterior side. So what you have here tries to depict it very well. So this is the mouth. It is located on the ventral side, over on the ventral axis. And what you have inside here is the pharynx. So how do these organisms feed, especially these carnivores? They have the power to protrude their pharynx from the pharyngeal region, so that it can go attached to the prey. When it attaches to the prey, there will be extrusion of enzymes. Then these enzymes shall break down the prey. Then there will be absorption of nutrients that have been broken down. That is why these organisms have both internal and external digestion. Then that is for the free living ones. Then for the parasitic ones, we have two forms. We have the flukes. These flukes, for them, they feed on body fluids and cell debris. In fact, sometimes they can even feed on the host's cells. Then we have tapeworms. So these tapeworms, for them, they have no digestive system. So they absorb nutrients direct from the host. Directly from the host. Then, still on the digestive system, we said it has a mouth, pharynx, and an intestine. And this intestine is highly branched. So this, what I have here is just an ultra structure. This is a branch of the intestine. This is also another branch of the intestine. This is also another branch of the intestine, and this is also another branch of the intestine. So the intestine is highly branched. In fact, it forms the basis for the division of members of class Tuberrelia, as we are going to be seeing. But one important thing to note, they have a blind gut made up of the mouth, the intestine, and the pharynx. However, researchers discovered that some species even have an anus, but those are the few. Most of them have a mouth, intestine, and the pharynx. The mouth is either located on the anterior side, especially in flukes, or what they call the trematodes. Then on the ventral side for planarians. Then they also have an intestine that is highly branched, and the pharynx in the pharyngeal region for feeding. Next, we are looking at the nervous system. So, earlier we said the sponges have some specialized type of cells, that act as the nerve cells. Then we saw a nerve net within the cnidarians. But here, since there is cephalization, they have a brain. But this brain is still rudimentary. It has cerebral ganglia. It's just a compilation of some ganglia. So what you have here, this is what you call the cerebral ganglia. On top of that, these some organisms also have a nerve plexus. This is a nerve net, as similar to that what we saw in cnidarians. Still, these ones have transverse nerves. These nerves that you're seeing, these ones that move from this side to this side, these are what you call the transverse nerves. Then they also have what we call ocelli. If not ocelli, they are called eye spots. Then see, they have statocysts. So the essence of statocysts, as we said earlier, is responsible for orientation of an organism with response to gravity. On top of that, they have what we call receptors, and chemoreceptors. So some of these organisms are hunters. So they need these receptors to detect for prey and to detect other changes in the environment. After having looked at the nervous system, we look at the reproductive system. The reproductive system. Reproductive system. So most of these organisms are monoecious. 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 And being monoecious means that they have both the male and female reproductive parts. However, they exhibit cross fertilization. Exhibit cross fertilization. That is number 1. Then some organisms have what we call a genital pore. Genital pore. Genital pore. Genital pore. This genital pore is where exchange of gametes takes place, as you are going to be seeing. Then see they have a trochophore larvae. Trochophore larvae. We say the trochophore larvae is a ciliated larvae. On top of that, some organisms, especially the parasitic ones, have complex life cycles. They have complex life cycles, as we are going to be seeing some of them.